Good afternoon from the Food Research Centre here in Fermoy, County Cork in the Republic of Ireland. Welcome to our third Infogest food digestion webinar. Today's webinar is on the topic of absorption models and intestinal barriers. And I would like to introduce the other panelists. If you can show yourself there, please. We have Mern Egan from our Food Research Centre, who's helping us with a webinar here. We have Linda Giblin from our own Food Research Centre here in Moore Park, and we have Adam Macierzanka, nice try Adam, from Gdańsk University. So without much delay, I would like to introduce the first speaker. We usually have two speakers. The first one today is Linda, and she will give a nice overview of the absorption models and the intestinal barrier. Linda is a senior scientist here in Teagasc. Her research interests are food bioactives and the gut barrier. She's the leader of one of the work groups, work group 3 on intestinal barrier models. She's also a leader in the EU cost action that focus on food for the older adults, which is NUT Redox. And last but not least, she is an associate editor for the Journal of Functional Food. So I hand over to Linda. All talks have been pre-recorded, thankfully, because we are still on the conservative side there. And subsequently, you can find the recorded webinar on the YouTube channel, and I show that to you later. Okay? Very good. Over to you, Linda.
Okay, so thank you for that introduction. And so let's start on food digestion and the gut barrier. So this webinar is really going to divide it into 4 sections. There's the gut barrier, the in vitro models, the ex vivo models, and some data from our lab. So in the last webinar, it focused on food digestion. So the mouth for mastication and mixing, the stomach for digestive enzymes, that very low acidic pH, the peristaltic waves, the small intestine, which has the addition of bile acids, the pancreatic digestive enzymes, that neutral pH, and of course the brush border enzymes. And then on to the lower gut, which is the cecum and colon, which has the gut microbiota. The bacterial metabolites are all produced, the short chain fatty acids, the vitamins are taken up, and water is retained. But in this webinar, I'm going to talk about the small intestine, because that is the main site for food absorption. And there are 3 sections: the duodenum, which is closest to the stomach, the jejunum, and the ileum, which is closest to the colon. And this tube is 6 meters in length, and it has a surface area of up to 250 meters squared because it has villi. This is what it looks like. All these villi protruding into the gut lumen. So inside the tube, it looks like this, millions and millions and millions of villi. And these villi in turn have all hair-like projections. And these villi differ in length and width from the duodenum all the way down to the ileum. And here is H&E staining to show you the differences that occur from the duodenum to the ileum. And by the time they get to the ileum, there are also patches are quite abundant in the ileum, which have an immune function in the gut. And these are not present in the duodenum. So let's talk about this villi. This villi is the gut barrier. It has a single layer of cells, and each cell has a tight junction with each other. And food has to pass through that single layer of cells to get to the blood system. So the blood capillaries will absorb the glucose and the amino acids, and the lymphatic vessels will absorb the fatty acids and the glycerol. But this single layer of cells is not just one cell type, but it's actually made up of 4 different cell types. The first one are the enterocytes, and they are the absorptive cells, and they have they're the most abundant by far, and they have all these microvilli. Next is the goblet cells, and these are the mucus cells. And I think Andrew showed you when he swallowed the camera in the last webinar, all the mucus that's produced in the gut. So these are quite common cells as well in that single layer. Then you have the endocrine cells, and these produce hormones. They are sparsely populated through the gut barrier, but they're very important. They produce these satiety hormones that make us feel full, and of course the hunger hormone that makes us start eating our food. And then we have the Paneth cells, which produce digestive enzymes and antimicrobial peptides. And the really interesting thing about the villus is that the enterocytes, the goblet, and the endocrine cells, they migrate up to the tip as they age. And by the time they get to the tip of the villi, they just die by apoptosis. And the Paneth cells, on the other hand, migrate down towards the crypts, and they're renewed the whole villus is renewed every 5 to 7 days during our lifetime, every 5 to 7 days. So the top of the villus could be regarded as the old part, and the crypts were regarded as the young villi. So how is food transported across the gut barrier? Well, if we talk about food digestion first, obviously the proteins go into amino acids, into small peptides, di and tripeptides, the fats into fatty acids, the carbohydrates into monosaccharides and disaccharides. So the gut lumen has all of this mixture of free amino acids, fatty acids, monosaccharides. But it has to be transported across that single layer of cells, that gut barrier, to get to the bloodstream. So how does it do that? There is paracellular transport, which is through those tight junctions. There is transcellular transport, which is a passive diffusion system through the cells. And then there's mediated transport, which is vesicle or carrier, where the food digested is literally brought up across the cells. It's transported, actively transported across the cells to the blood. So what are the in vitro models that are present that we can use? So the first one and the most commonly used one are the CACO2 cells, and these are derived from a human colorectal carcinoma, and you can grow them in transwell plates for 21 days, and they'll actually differentiate into enterocytes, and these cells will be polarized, and they'll have tight junctions, and they'll have an apical brush border. And you can measure these tight junctions by running an electrical current through them and measuring the resistance, the electrical resistance, and that's called the transepithelial electrical resistance. And it should be greater than 500 ohms centimeters squared. And then you know you have a tight junction formed. And that usually occurs around day 20, day 21. You can also measure permeability across these monolayers by using things like luciferase yellow or sodium fluorescein, applying it to the apical side and then measuring how much comes through on the basolateral side. But not only are they colon cell lines, but they're also a single cell type. So nowadays, we usually mix them with a different cell called the HD29-MTX, which are goblet cells, and they produce mucus. And so this is a co-culture in these transwell plates, and usually we mix them at a ratio of 3:1, CACO2 to HD29, but they can go up to about 9:1. So this is the most common co-culture that's used as a model of the intestinal barrier. But there are lots of other alternative cell lines that you could consider using as well to form your gut barrier. And they really depend on the animal. There are several available from young piglets or from an adult boar. There's a few from the human and the rat. Also, it will depend on the location that they have been harvested from, the jejunum, the ileum, the colon or the duodenum, and indeed whether they've been derived from a cancerous cell. And what I would say is that all these cell line barriers do have limitations with real life, and you have to consider your primary research question. Are you trying to compare and contrast your results with your food component to others? And if so, I would recommend using the CACO2 HD29 since that's routinely used by a lot of scientists. But maybe you're looking for immune response, and then maybe the IPIC-J2 is much more suitable. There are other cells that cell lines that mimic the cell types in the gut barrier. They do not form the barrier, and these would be enteroendocrine cell lines. So if you're looking for a response of a satiety response of your food, then you could look at these cell lines, STC1, NCIH716, or GluTag, but they don't form gut barriers. They're specifically enteroendocrine cell lines. In terms of ex vivo models, well, I suppose the one that is most commonly used and has been around for a long, long time now are the Ussing chambers. And really, this is where you take an intestinal tissue and you immediately apply it to a reservoir to form your barrier between the apical and the basolateral. And these have been multiplexed up to about 10 reservoirs, but they're still relatively low throughput. And the limitation, of course, is that the availability of healthy human samples. So usually you have to use pig samples and rat samples, and you have to do the experiment straight away once you've collected the tissue. So next, then, there is the intestinal organoids, and these are self-sustaining mini-guts. And so what they really start with is that intestinal tissue is taken from the human, the rat, or the pig, and from there you isolate the crypts. And these crypts can be fully matured then over a 7-day period until finally they become self-sustaining mini-guts. So it's a very exciting part of research, but the one issue it has with food digesta is that these organoids face inwards, so they have an inward orientation, which means that the apical side is relatively inaccessible. So what people are doing now is trying to create these organoids, but having an outward orientation, so we can use them much more readily for food digestion experiments. Okay, so let's put together all these methodologies. So how do you use this work in the lab? So you take the food that you're interested in, the food component or the food matrix, and you perform an in vitro digestion, either a static digestion, semi-dynamic, or dynamic digestion. In parallel, you grow your CACA2 HD29 co-cultures in these transwell plates. You allow them to differentiate for 21 days. You check your tight junction formation by your TER values, making sure it's greater than 500 ohms centimeter squared. Then you apply your digesta for 2 to 4 hours to the apical side, and you collect the basolateral and look for the food components that were permeable across this monolayer. So now I'm just going to talk to you about some of the data from our lab and how we've used all of these methodologies. So what we wanted to find out here was whether processing would affect the peptides that were permeable across the CACA2 monolayers. So we made infant milk formula, and we made it by 2 different methods in the pilot plant. One by high temperature, which is what is used routinely in infant formula manufacture. And the other method, the alternative method, was making it by cascade membrane filtration. And both of the IMF products that were made were microbially safe and met all the standards required. We then took each of these products and we digested it using a static Infogest protocol for the infant gut. And that has a higher gastric pH, it has a shorter transit time, and it had lower enzyme activity than the adult protocol. So here we take the infant milk formula, and this is produced by the high temperature processing, and we have it digested. So we add it to our CACA2 monolayer and for 2 to 4 hours, and we collect the basolateral and look at the peptides that are coming through that are permeable across that monolayer. And this is a pie chart of the type of peptides that we picked up: 30 peptides from beta-casein, 7 from kappa-casein, 21 for beta-lactoglobulin. If we repeat that with IMF that was made by cascade membrane filtration, we find that we get a different profile of peptides. We get 42 peptides from beta-casein, 34 from beta-lactoglobulin. And in fact, if we look at these individual peptides, we find that there are several unique peptides crossing those CACO2 monolayers, which are dependent on the processing type. So there are a lot of peptides that are in common, but there are also some unique peptides crossing the CACO2 monolayers. So processing does affect the peptides that are permeable across these barriers. Another research activity we were working on was whey proteins. We know that whey protein promotes muscle health. It does that because it's an easily digested protein. It's a high quality protein. It delivers essential amino acids and indeed branched chain amino acids to muscles. So athletes love it. And they use the whey protein bars or whey protein isolate. And in fact, it could also help delay frailty in the older person. But what we wanted to do was, we wanted to find out, were there peptides capable of passing the gut barrier, which would also promote muscle health? So to do that, we took whey protein isolate that is commercially available, and we performed an in vitro digestion. In this case, it was a static adult protocol. And we grew our CACO2 HT29 co-cultures for 21 days, make sure they had tight junctions. And then we added this gastrointestinal digested whey protein isolate to the apical side. And we collected, again, the basolateral and performed peptide analysis of the basolateral. And this just gives you an overview of what we got. So at the very top is all the peptides that we identified in the apical side. And then at the bottom is all the peptides that we identified in the basolateral that had obviously were permeable across that monolayer, that CACO2 HT29 monolayer. And several of these peptides we ran through a software analysis and were found to be bioactive, or they were predicted to be bioactive. They were predicted to be opioid peptides or antioxidant peptides or antibacterial peptides. So we thought, let's have a look at these. Let's synthesize some of these peptides. Okay, so we selected some of these whey peptides that we know had come through the CACO2 monolayers, and we synthesized them. And in parallel, we grew muscle cells in the lab. And what we did was we stressed these muscle cells, and that's the red column there, that's a stressed muscle cell, and we're going to give it an arbitrary unit of 100% stressed. And this would be modeling post-exercise or indeed a very old muscle. And if we give it N-acetylcysteine, which is the purple column, that's a positive control. It will always reduce stress. So it's reducing the stress from 100 down to about 60%. There's your purple column. What about the peptides that we have synthesized? Well, we find that if we add these peptides to muscle cells, we actually also get a reduction in stress from the 100% down to 40 to 60% with at least 5 of these peptides. One of these peptides does not help the stress, but 5 of them do. So that tells us that there are bioactive peptides in whey which will reduce stress in muscle cells. So really what I've tried to do in this webinar was talk to you about the methodologies we have for combining digestion with bioavailability and with bioactivity for different life stages, for the older consumer, for the infant, and indeed for the athlete. Within Infogest, there is a working group that focuses primarily on intestinal barrier models. And this is made up of 60 scientists from 14 different countries from 26 research institutions. And the objective of this working group is really to put forward a series of recommendations of how to treat these intestinal barrier models with food that's digested. And then how to compare and contrast the data from these cellular models with in vivo data. So this working group 3 then is divided up into 7 sub-activities, I would say, subgroups. And so I apologize in advance now for my poor pronunciation of names. But Alina Kondrashina leads the group on how to deal with food digesta before you put it down on these monolayers, how to detoxify it, how to inactivate the enzymes. Do you add protease inhibitors? Do you inactivate by temperature? Gianfranco Mamon then works on brush border enzyme activity, and that is really how to incorporate this enzyme activity into digestion protocols, but also how to protect it on these monolayers when they're incubated with food digester. Shauna Bastianette then looks at allergy and inflammation and how our in vitro models represent real life. Beatrice Morales then looks at permeability ring trial, where we look at how the interlab variation when you're doing the same experiment with similar protocols or the same protocols. Lydia Thomas then looks at colonic fermentation and adds that layer of complexity of putting colonic fermentates down on monolayers. And then Elena Arnez looks at cellular bioassays and the wide variety of cellular bioassays that are out there to look at bioactivity of these food components. And then Brig Graf looks at the in vitro versus the in vivo data. Does it really truly match what's happening in real life? So if any of the people listening are interested in our working group, please contact me by email and we'd be more than happy to have more members. So I think that's it. Thank you very much for your attention, and over to you guys.
Excellent talk, Linda. Thanks for that. It was a good overview. So we have time for a number of questions, and I might start because I get this question quite often. I got it by email on Monday from Greece, from Elena Dalaka, and she asked a very common question again. When you do the in vitro digestion of the food, how do you prepare the digester for cell cultures? Because obviously there are a number of inhibitors and toxic elements in the digester.
Yeah, Andre, that's a very good question. So the problem with the food digester is that it has active digestive enzymes. So you have to find a way of getting rid of them. So there's 5 main ways of getting rid of them. You inhibit them by using protease inhibitors, you heat and activate them, you use a molecular weight cutoff to remove them, you dilute it, or you use media and FBS. So if we start with the protease inhibitors, what happens there is, of course, that if you use protease inhibitors, they're going to also affect your brush border activity on your monolayers, and they're also going to interact with the way the cell functions. If you use heat and activation, well, for us, we can't use it if we're looking at a processing, the effect of processing on food digestion. We just can't bring in another thermal step. If you look at molecular weight cutoffs, that's fine if you're dealing with dipeptides and tripeptides, but it's a big deal if you're looking at glycosylated proteins or bigger components, food bioactives. If you dilute it, your instrumentation may not be able to detect anything in the basolateral. And then finally, if you use media and FBS, you may mask any health benefit you have from your food digester because the cells are getting plenty of nourishment from the media. So, so that's 5 things you can do, but there's no solutions in any of that. And what people normally do is they use a mix of those things. And we have a working group looking directly on that. Elena Kondrashina is looking at that to try and get kind of consensus about what we should and should not be doing. But it's not solved, but there are the options at present.
Yeah, very good. So if you have, if anybody else has a question regarding the detoxification, you can send an email to Linda, because there's a whole work group just dealing with that, because it's case by case really what
yeah. Yeah.
Another question, Linda, is it physiologically relevant to use cancer cells to mimic the gut barrier ex vivo?
Okay, that's a question we get for everything we do with cell lines, really. So thank you, Muireann. So basically, it's not, they're cancer cells. So the reason why people use cancer cells, and certainly the reason why we use it I can talk about the gut barrier in a minute, but the reason why we use cancer cells they're immortalized, which means that they grow independently and they grow easily outside the body. The reason why we specifically use them in gut barrier is because they've been traditionally used by the pharma industry for drugs and drug permeability. So it allows us to compare and contrast our food digester. There are primary cell lines out there, but if you do primary cells, you're taking tissue, you're growing them for a very limited number of times because they're self-limiting, and then you have a lot of experimental variation from day to day. So I did mention one of the cell lines there is the IPEC-J2, which is a primary immortalized cell line. So I would imagine that more and more people will start using that, but again, it's a little bit more tricky to grow than the CACA2's HD29s. So I hope that answers the question.
Very good. I have another question from Isidra Recio, and she asked, could you comment on the positive and negative control that we used on CACA2 transport studies to ensure monolayer integrity?
Okay, so thanks, Isidra. So basically, the first thing you do, of course, is these transepithelial electrical resistance, which is to run the electrical current through and measure resistance to make sure you have tight junctions. And then you put your food digester down on these monolayers. So the first really important control is to have a digester control. So that's where you've carried out the whole digester, but it has no food in it. And the importance of that is you need to know what is your background noise on those monolayers. Luciferase yellow is used to determine that you have tight junctions because its transport is through the paracellular, and then fluorescein, I think its transport is more active, so that's another way of looking to make sure your transport systems are up and running. But the digester control is the most important thing you will run.
Okay, very good. That answered it. We have a number of questions. We have time for one more. During the talk, Linda might answer the other questions, or she types it. If we don't have time during the 1 hour, we probably answer all the questions afterwards. Okay. So there's one more from Diana Oliveira. Asks if this model can be used to study other nutrients beside peptides. If I want to check what nutrients are being absorbed, for example.
Oh yeah, of course. I just use, that's just my area of expertise. So the CACA2 monolayers have been used by lots of different foods. So carotenoids to even uptake of vitamins and minerals, much more complex food matrices for fatty acids and all of those things. It's very commonly used. As I said, we brought it from the pharma industry. But it's the interaction between the food digester and the CACA2 that's making it particularly complex for us. And of course, with colonic fermentations, you're looking at bacterial metabolites like short-chain fatty acids and things like that. So,
very good, very good. Yeah, thank you, Linda. I think that was excellent. So we might move on to the next speaker. Adam, can you show yourself? Excellent. So Adam Macierzanka is a professor at the Gdańsk University of Technology. He's also the head of the Department of Colloids and Lipid Science. His research expertise and interest is in the functionality of food colloids and the interaction between food structure and the human digestive system. And most recently, his research focused on the validation of animal materials such as bile and intestinal mucus for the use of human relevant in vitro digestion models. Okay, so I hand over to Adam. Again, the talk is pre-recorded and we have a live question and answer session. If you have any questions, type them into the question and answer, preferably during the talk, so we have a little bit of time to go through them. Okay.
Hello, this is Adam Macierzanka from Gdańsk University of Technology. I have a pleasure to give you a brief overview on the role of mucus barrier in the transport of nutrients and particles in the small intestine. The mucus in the small intestine covers the surface of mucosa. It's a selective barrier. It can prevent the epithelium from direct contact with microorganisms, including pathogens. It also lubricates the passage of food during the digestion in the gut. And of course, all the nutrients which are released from the food during the digestion would have to be exposed to the mucus on their way to the epithelium. So all the nutrients and bioactives which are released would have to cross the mucus layer in order to get absorbed by the underlying epithelium. The thickness of the mucus layer in the stomach and also in the colon is considered to be larger than the one in the small intestine. Here, it can differ between duodenum, jejunum, and ileum. It can also fluctuate due to, for example, removal of mucus with the food during the peristaltic motion. This image shows a cross-section of the proximal small intestine in the pig. In the dark colors, you can see the mucus layer, which separates the mucosal tissue from the lumen. And when you look at the specimen under the microscope, and here the mucus has been stained for mucins in green, you can see that the mucins, which are one of the major components of mucus, fill the spaces between villi. They also cover the tips of villi in the form of a layer and aggregates. So the mucins have long been considered the major macromolecular and gel-forming component of mucus. Mucins are glycoproteins, and in the small intestine, but also in the large intestine, the main secreted mucin is MAC2 mucin. One of the most distinctive features of mucins is the protein backbone with all linked oligosaccharides. This highly glycated region is also called the PTS-rich region. Here the glycation is shown in green color, and in the Golgi of the goblet cells in the epithelium there is formation of dimers, trimers, oligomers of mucins, and eventually the mucins are assembled in the form of granules in the goblet cells. The image on the right-hand side is a confocal microscopy of a single villus in the small intestine. Here we can see enterocytes responsible for taking up nutrients. The surface of villus is covered in mucus. So again, the mucins in the mucus have been stained in green color, so they are still either in the form of granules inside the goblet cells or have already been secreted and contribute to the mucus layer. Here is an atomic force microscopy of MAC2 mucin granules that already expanded to some extent. Inside the granule, the mucins are packed due to calcium-dependent cross-linking, and under physiological conditions of the gut, the expansion of the granule requires an increase in pH and also the removal of calcium. Eventually the mucins would form a layered network with well-defined pores in the mucus, and here we have an example of the mucin network in the mucus removed from the small intestine of the mouse. But very similar organization of the mucin network can be also found in the small intestine of the pig and in humans as well. When you look at the small intestinal mucus under the confocal microscope, you can see that its structure and composition can differ between different locations in the tissue. Here we have again a cross-section of the proximal small intestine in the pig. The image B shows the magnified view of this fragment of the specimen with the connective tissue and villi. And then if we go even further, the location C within the specimen, this is the mucus located between adjacent villi. So the green color represents the mucins in the mucus, whereas the red color shows the nuclei of epithelial cells and the cells of the connective tissue. So when we compare this mucus with the mucus layer, which is sitting on the top of a villus, this is position D, we can see that within the mucus layer, apart from the mucins, there are also particles of DNA embedded. And the DNA particles can be also found in the aggregates of the mucus, which are exposed to the lumen. The coexistence of the extracellular DNA and the mucin in the mucus is even more obvious from this image, showing the tip of a villus with the protective mucus layer. Here we have a magnified view, and in the image C we can see the network of the mucin, whereas in the image D we can only see the DNA in the mucus layer. And the DNA is at various stages of degradation, from almost intact nuclei to the network of small fragments. So where does the DNA come from? The intestinal epithelium undergoes a continuous self-renewal, and the new cells which are produced in the crypts would differentiate and migrate within several days to the villi tips. Eventually they would be removed from the tissue. This is a physiological process, and as a result of it, there is accumulation of the dead cells and the nucleic acids from these cells within the mucus located around villi tips. The contribution of the DNA to the microstructure and permeability of mucus can be evaluated experimentally using, for example, a multiple particle tracking method. Here we have a schematic representation of the mucus located in between villi and also on the top of the tissue. On the left-hand side, we have X and Y images showing the mucus at various locations in the tissue. So here, for example, this is the mucus on the top of the tissue, and then in B, C, D, and so on, we look at the mucus which is located deeper and deeper in between adjacent villi. In this experiment, the mucus in all these various locations was incubated with probe particles, and then the movement, the diffusion of particles was evaluated experimentally. This is a video clip showing movement of particles in the small intestinal mucus. The principle of the MPT method is that we can track the position of individual hundreds or even thousands of particles in time. And then from the trajectories we can define some parameters of the microenvironments the particles were exposed to. So here, for example, we can see particles which were immobilized by the mucus structure, whereas some other particles were able to diffuse in the other locations of the mucus. This is how raw MPT data usually look like. So we have a broad range of distances covered by individual particles over the timescale of experiment. And this is because some of the particles could only diffuse in very confined spaces or were completely immobilized by the mucus, whereas other particles were able to diffuse in different locations of the mucus. And here we have examples of the tracks of diffusing particles. The distance data is usually used to calculate mean square displacement, and from the data obtained from individual particles we can define the families of diffusive particles and also the particles which were immobilized by the mucus, the subdiffusive particles. In our experiment, the mucus which was located on the surface of the tissue was found much less permeable to particles than the mucus located 40 or 80 microns below the top, so between the villi. And this significant difference might have been due to the accumulation of the DNA in the mucus around the villi tips. In experimental practice, the mucus removed from the tissue is usually used. In this collected ex-vivo mucus, the coexistence of the mucin network and also the DNA was seen. In order to confirm the contribution of the DNA to the microstructure, but also to the permeability of the mucus, the DNA was selectively hydrolyzed with DNase. The DNase treatment caused a very significant improvement in the number of diffusive particles. More than 60% of the particles in the treated mucus were able to diffuse, whereas in the control mucus, which has not been treated with the DNase, the particles were immobilized by the mucus structure. In any experiment where spherical particles with well-defined sizes express free diffusion in mucus, the MSD data can be used to calculate the diffusion coefficient of each and every particle diffusing in the mucus. And then the diffusion coefficients can be used to calculate the microviscosity of mucus. Here we have an example of experiment where particles were used to probe heterogeneity of mucus. The particles were incubated with the biocells in order to prevent mucoadhesion, and then they were exposed to the mucus. Using the diffusion data, it was shown that the small intestinal mucus is very heterogeneous, with the viscosity ranging from the viscosity of water to the viscosity several orders of magnitude larger than the viscosity of water. Translocation of particles in mucus is much more complex if they differ in size or in the surface chemistry. There is a concept of size filtering, which assumes that particles smaller than the mesh size of mucus are allowed to pass while larger particles are rejected. There is also a contrasting concept of interaction filtering, which is based on the strength of interaction between particles and the mucus polymer network, so the weakly interacting particles are not retained in the mucus and allowed to diffuse. A good example of formation of a very complex dispersion of particles is the small intestinal digestion of lipids. Here, large droplets of lipids are emulsified with bile salts to the smaller droplets. And finally, there is a formation of a range of mixed micelles, which are composed of the bile salts, phospholipids, and the products of the lipolysis. This is how it looks under the microscope. Here, a single droplet of oil was incubated under simulated small intestinal conditions with pancreatic lipase and bile salts. And due to the action of the enzyme, the triglycerides were digested to fatty acids and monoglycerides at the surface of the droplet. The products of lipolysis and bile salts form spontaneously a mixture of particles that range from large spherical aggregates to smaller ones, and also to very small mixed micelles, which of course were too small to be seen under this magnification. In the next experiment, I want to describe a similar digesta dispersion was produced from a commercially available yogurt. The yogurt was digested in dynamic in vitro model of gastric, followed by small intestinal digestion that simulated conditions of adult humans. And samples of digesta were removed from the small intestinal compartment after 1, 2, 3, and 4 hours of digestion. And then the penetration of digesta into porcine small intestinal mucus was measured using time-lapse confocal microscopy. The mucus was placed in optical cell under the microscope on one side of the cell, and then on the other side of the cell, the digesta stain for lipids was placed, and the fluorescence profile for lipids was recorded at time 0. The time-lapse microscopy was used to look at the penetration of digesta into the mucus. And after time 0 and then 10 minutes, 20, 30, 60, up to 90 minutes, we can see a progressive penetration of lipids from digesta into the mucus layer. And of course, for every time point of this experiment, the fluorescence profiles for lipids were recorded. And then from the profiles, it was possible to calculate effective diffusion coefficients for digesta in the mucus. This is how the final data look like. The diffusion coefficient of digesta was reduced by about 70% over the first 30 minutes of penetration into the mucus, and then it was constant until the end of the experiment. The reduction might have been due to the clogging of pores and channels inside the surface region of the mucus by the largest particles of digesta. This can suggest that the penetration inside the mucus can only continue after the particles have been reduced to a certain size. It can also suggest that under physiological condition of the gut, the lipolysis can continue after the lipids were moved from the lumen inside the mucus layer on their way to the epithelium. A similar filtering effect of mucus was observed before, when sodium alginate was penetrating inside the mucus layer. The incorporation of alginate was used in order to increase the overall viscosity of mucus. And then the diffusion coefficient of lipid digesta inside the mucus was measured, and especially for the lipid digesta produced after 120 minutes of duodenal conditions. The diffusion coefficient of lipids was reduced significantly relative to the control mucus. This can suggest that such a modification of mucus layer can be a promising strategy in reducing lipid uptake in the small intestine. Experiments on penetration of digesta into the mucus layer can also be used to answer the question whether the porcine mucus, which is quite easy to obtain and often used in transport experiments, is actually a good substitute of a difficult to obtain human mucus for human relevant studies. In order to answer this question, we used porcine mucus as a reference material. We collected mucus from the jejunum and the ileum of adult pigs. We also included jejunal mucus collected from piglets in the study. And then the mucus from all these sources was exposed to the lipid digesta, and we measured the penetration of digesta inside the mucus. We also included human mucus in the study. The mucus was aspirated during the standard colonoscopy procedure from a number of patients at a clinical hospital. As a part of any colonoscopy procedure, the distal part of the ileum is intubated with the colonoscope, and this is where the aspiration of mucus was done for our study. Calculations of diffusion coefficients of digesta in the mucus collected from the adult pig jejunum and also the pig ileum showed that there was no difference in the penetrability of the mucus collected from these two different anatomical locations in the adult pig small intestine. More importantly, the digesta penetrability of the human mucus was very similar to the one observed for the adult pig mucus samples. So the diffusion coefficient of digesta in the human mucus was not statistically different from the diffusion coefficients recorded for the digesta in the adult pig mucus samples. The only mucus which was much more penetrable to lipid digesta was the piglet mucus, and this might have been because of the different structural organization of this mucus, which was more heterogeneous and fragmented. This was probably because of the different kinetics of the mucin secretion and also the cellular turnover in the neonatal gut. And finally, a couple of conclusions for this part of the presentation. The small intestinal mucus collected from adult pigs seems to be a good substitute for a difficult to obtain human mucus in human-relevant studies on colloidal transport in the gut. And also, properties of mucus seem to differ quite significantly between different age groups. We saw the difference between adult pigs and piglets here, but it also suggests a similar difference can be observed between adult humans and infants. Thank you for watching.
Thank you, Adam, for excellent talk. So we have time for some questions, and I might start. Adam, as you know, in the Infogest network, we have been working on standardizing digestion methods, and you know, at times quite difficult because we want to use established methods. They have to be rock solid, reproducible, and so forth. And we've already seen what is on the horizon, meaning population groups, infants and elderly. We have also talked about brush border enzymes. There wasn't any consensus when we wrote the last paper, but it's certainly on the horizon. The use of mucin or mucus, is that something for the foreseeable future that we can use to add to in vitro digestion to make it more accurate?
I think so. Thank you for the question first of all. Especially, you know, over the last years, when we look at the mucus and the effect of the mucus, I mean, it starts to be obvious that it can really slow down, or it can really regulate the transfer of nutrients or particles. And you know, 10, 15 years ago, when we started to look at the effect of the mucus, and it was back in Norwich, and the research was initiated by Alan Mackey, then we focused on the mucus which was collected from the tissue. So we look at the effect of things like, you know, biocells, and whether you can actually freeze the mucus and use it later. And then, you know, more recently, we'll look at whether the mucus from pigs is actually a good model for the human situation. So, and now we know the answer. You know, it is, and now. Also, it's getting more obvious that, you know, that the structure, the native structure of the mucus in the tissue is very important. So we move from actually using the mucus which was removed from the tissue to the mucus which is still in place, which we call a native mucus. So I remember one of the comments which was made by Linda during her presentation, and she said that one of the limitations of the using chambers at the moment is actually the limitation in the availability of the human tissue samples. And then the last slides that I presented in my talk actually showed that the porcine mucus is a good substitute for the human sample. So I would imagine that, you know, at some point we can use, especially using chambers, and use porcine tissue as a replacement for the, as substitute for the human tissue, because then of course we can preserve the native architecture of the mucus. And as I tried to convince you, that DNA in the tissue, in some parts of the mucus, is very important in terms of the overall kinetics of the penetration by digesta, not by, you know, trace particles, by actually something we call a physiological food digester. So yes, you know, I would imagine that at some point, especially using chambers, will be part of the standard protocol that would, you know, that would combine digestion and the transport part as well.
Very good, very good.
Okay, Adam, so next question is, in the example of the yogurt digestion penetration into the small intestine mucus, which model was used? Was it an ex-vivo tissue model?
No, it wasn't tissue, it was ex-vivo mucus. So the mucus was actually removed from the tissue. And when we remove the mucus, you know, the layer of the mucus you obtain, you get, is the one which contains the DNA. So this is that part of the mucus which is exposed to the lumen. So we believe this is the first part of the mucus which is directly exposed to the digesta. And then we use mucus from 4 different sources. We focus on comparing the human versus pig, and it was for the adult organism. And it was the mucus from the ileum. Then we also compared the mucus from the jejunum and also from the ileum for the pig. And then finally, we compared different age groups and the jejunal mucus from piglets and the adult pigs as well. So we just did 3 comparisons in one study.
Very good. We have 2 short questions. So Adam, if you can answer it quickly as well. One is the effect of shear. How does it affect the mucin and the absorption? And then there's a question from Tim Lambers. Do the methods you have used for tracking transport also allow the setting to study the turnover of mucus? Okay, the particle tracking for turnover, and the first one was the shear.
The first question about the shear, of course the shear would affect the mucus, and of course it would affect the overall kinetics of the transfer of nutrients towards the epithelium. And actually there was a number of studies looking at the thickness of the mucus. When you compare them, the thickness of the mucus in the ileum or even the jejunum, and the studies were done on rats, as far as I remember, you know, the mucus can actually be reduced between, as far as I remember, 7 to 20 times, you know, after the food actually moved, you know, along the small intestine. So yes, the shear would affect, especially the thickness of the mucus, that's for sure. And the other question was about using the particle tracking.
Yeah, the particle tracking, if you can use it for the turnover of the mucus.
You can certainly use it for looking at the turnover of the tissue, because, you know, the result of the turnover would be accumulation of, for example, the DNA in some parts of the mucus. And one of the slides was actually dealing with that situation. So we look exactly this method, the particle tracking, to look at different Z positions of the mucus between villi and also on the top of the mucus layer, and we saw significant differences. So the short answer would be yes, but because there are many aspects you can look at.
Okay, very good, very good. So I would like to thank both speakers, Linda and Adam, for a nice talk and a good question and answer session. As I mentioned earlier, all questions, we try to answer all questions even afterwards. So you probably get an email afterwards. If not, just send us an email, just a reminder, a gentle reminder, and we will answer your question. Okay, thank you both. I just have 2 or 3 slides before we finish at 3 o'clock Irish time. Give me 1 second. The wrong slide, but that's fine. Here we go. So just to mention that we have this webinar. This webinar will take place every first Wednesday of each month, 2 o'clock Irish time. So if you Google Dublin, you know that's the Irish time. The next webinar will be 2nd of December, 6th of January, 3rd of February. Usually we have 2 speakers and we try to stick to one hour. We will publish all webinars on YouTube afterwards. So if you Google Infogest webinar, you will eventually get there. We have in 2 locations, one on our own website, Teagasc, and the other one on the Infogest YouTube channel. If you want to be part of Infogest and want to get an email update, please send an email to Didier Dupont, who is the chair of Infogest, or Nathalie Lemar. I also created a Infogest group on LinkedIn. So if you go to LinkedIn, type Infogest and follow, you will get the regular updates. You can post yourself as well. So I just gave an example down here. If you want to post my last brilliant paper on such and such, and then type @ Infogest, you will get the notification. So other people will get the notification. I think at the moment we have 300 or 400, quite a few follow the Infogest channel. So you get news, and it's also a way for you to publicize your research. Just a bit of a background, the Infogest is an international network. The chair is Didier Dupont and the vice chair is Alan Mackey. We have several working groups. We have started the first few webinars. First 2 webinars were on these groups here, in vitro models and food interaction with digestion. Today's webinar was on the absorption models. The next webinar will be on digestive lipase and lipid digestion. That's in December. In January, we look at amylase in the starch or carbohydrate digestion. And in February, we look at in silico models. So if you want to present at one of these webinars, please send us an email, either myself or Didier or the work group leaders, because in the end, it's the work group leaders that will select the speakers, and we will publicize that either by email or on the Infogest, on the LinkedIn page. Okay, so that's my last slide. We were about 180 people live, and usually we have a few hundred people that look at the YouTube channel, so it's a good way of really publicizing your research. Okay, so see you next time in December. Goodbye and slán.